So microbes are everywhere, but we can't see them with the naked eye. So my lab aims to make them visible, to really study them at the molecular detail. So in order to study microbes in an intact state, we use a method called cryo-EM, which allows us to study the microbes in a flash-frozen state at high resolution. So once the cells are frozen, we can take images from all different angles and reconstruct them in the computer. And then we can study the microbes in 3D at macromolecular resolution. So my lab is especially interested in the chemoreceptor rays of the bacteria. These are sensory rays that are located at the cell poles. They're composed of thousands of receptors that cluster together in honeycomb-like arrays that then help the cells navigate their environment. So in order to address our research questions, we collaborate with a lot of different scientists from different fields, such as geneticists, biochemists, structural biologists, or bioinformaticians from the Netherlands and all around the world. And the aim is really to combine all these disciplines to answer fundamental questions that in turn will allow us to harness bacteria, for example, to clean wastewater or to combat pathogens.
So my research focuses on how plants deal with stress. And the very first step of how plants perceive stress is something that we really don't understand. So that's still a black box that we are investigating. And then we focus in particular on the root system, as that is the first tissue to encounter the stress. And what we're trying to understand is what makes some plants resilient. How do some plants get it right while other plants are really sensitive to salt? Well, we live on a planet with a changing climate, and this changing climate means increased prolonged periods of drought, and this leads not only to water deficit for plants but also increased soil salinization, because the water evaporates but the soil stays behind polluted with salts. Well, I think one of the major challenges in plant biology is to understand how plants change their development in response to a complex environment, and for that we really need multidisciplinary research. Not only focus on a single stress but on multiple stresses, use mathematical models and also understand the field situation. I think the technical challenges at this point lie in the understanding responses at the tissue or subtissue level, and at the same time understanding what they mean for the physiology of the entire plant. So in our research we don't only focus on model organisms, but we really want to expand to crops and to field situations. So the example is the collaboration with the Rice Research Institute in the Philippines, which is interesting both from applied science but also fundamental science, because it helps us understand how plants deal with a complex environment outside.
My research is about the effect of antidepressants in pregnant moms and how that affects the offspring. It's very interdisciplinary, and therefore I collaborate with many disciplines. Serotonin is affecting, for instance, the placenta. We are studying with RNA sequencing which different genes are actually affected by antidepressants. And because we want to see whether this is evolutionarily conserved, we study this in fish, rat, and in human placentas. So serotonin is approximately for 90% in our gut, and all these microbes that are in our gut, they are affecting brain development in the child as well. So we are studying how antidepressants in the mom are actually affecting brain development in the offspring. So when a mom used antidepressants during pregnancy, we see that there's a reduced play behavior in the offspring, and this continues until adulthood for males especially. And we are interested also in the brains, which genes are altered because of that. So one of the possibilities is that the brain develops faster after use of antidepressants, and we saw that the myelination of the gene expression and the methylation is altered in offspring that is treated with antidepressants. With our research, we hope to get insights into the mechanisms that are underlying the changes in the offspring after use of antidepressants in the mom. And this, in the end, hopefully will help the mother and the physician to decide whether to take antidepressants during pregnancy or not.
We investigate the distribution of migratory birds. So we want to understand why they go from one place to another. We also want to understand why their numbers change. This is a battle godwit. It flies from the Wadden Sea to Far Eastern Russia. Far Eastern Russia has their most rapid climate change. These godwits tell the story. We are the interpreters as scientists. They connect the dots. In order for these migratory birds to tell us the stories, we equip them with very small transmitters that communicate with the satellite network. Wherever they go in the world, we know their position and how they move from place to place. The birds I'm studying are only here in the Netherlands for a very brief time of the year. A very important time, but a brief time. So obviously we work with a worldwide network of people just as engaged with these birds as we are. Obviously, I care about migratory birds. I want to understand them. But what I also realize is that they have a lot to tell us about the conundrum of our small globe and the big universe. We're in trouble at the moment, and these birds can help us find a way out.